with fungi, oftentimes they do have distinct shapes. So if we're talking about a yeast, they often have an oval appearance, and then with things like molds and even the mycelium that make up the mushrooms, they typically have those long filaments of hyphae. There are some species of fungi that can kind of shapeshift, and in some environments they may look more like a yeast and be oval, and then in other environments they may look more like a mold and be a long filament. If a fungi has the ability to do that, we say they are dimorphic. Candida albicans is one that is considered dimorphic. In certain environments, it will appear as more of a yeast, and we can see this in this image. This is all Candida albicans here, but we can see here where it's in more of the yeast form, which are these ovals. But then we can also see here that it can exist in more of a mold-like form, as is represented in the septate hyphae present here. So if they are able to exist in both forms, we say they are dimorphic. This is similar to how bacteria can morph into different shapes depending on mutations and environmental characteristics. Although when bacteria go through this change, we say they are exhibiting pleomorphism. So same concept, just different terminology based on the organism type. The next thing is to talk about the fungal reproductive spores. We won't go through fungal reproduction, as that is a fairly complex process that could be an entire course in itself. If you ever take mycology, you will learn all about fungal reproduction. But for us, we just want to look at the different ways that the fungal spores or the reproductive units can be stored. So sometimes the fungal spores are stored inside of an encased membrane. This would be consistent with something like a mushroom. So it would be encased and then the spores would be inside. If that's the case, then the spores are known as sporangiospores. And if this encasing is broken in any way, those sporangiospores would be released, and then new growth would be generated wherever the spores would land. Candida are free spores, so they are spores that are not encased, and this is very common for mold. So if we look here, these little circular structures at the top here, these are all technically Candida spores. They're not encased in any type of membrane. So even something as simple as a gust of wind could dislodge these Candida spores and then cause them to be distributed in the environment and lead to new crops of mold development throughout the immediate environment. So if the spores are encased in a membrane, we would technically define them as sporangiospores. If they're free living and not encased, we would define them as being Candida. So if someone has a fungal infection, we say that they have a mycoses. And there's different classes of fungal mycoses. We'll learn specific infections later on in the infectious disease chapter. But for right now, we want to categorize fungal infections. So there are 4 different categories: superficial, subcutaneous, systemic, and opportunistic. So what you're probably most familiar with, because they're the most common, are the superficial fungal infections. These would be fungal infections that target any keratinized tissue, such as hair, skin, nails. So when we talk about things like jock itch and toenail fungus, well, those are examples of superficial fungal infections because they're targeting keratinized tissue. Then subcutaneous fungal infections, they occur deeper within the skin, below the keratinized tissue in the subcutaneous layer. Systemic fungal infections target any internal organ. So if you have a fungal infection in your brain, heart, kidneys, lungs, liver, wherever, it's considered a systemic fungal infection. And then opportunistic fungal infections only affect those individuals who are immunocompromised. So if someone is immunocompromised as a result of diabetes or cancer or taking immunosuppressant drugs for an organ transplant, and they get a opportunistic fungal infection, we would classify it as such. I have a few examples here at the bottom. So histoplasmosis is a fairly severe fungal infection in the lungs, which are an internal organ, so that would be classified as systemic. Fungal pneumonia in an AIDS patient. An AIDS patient would be severely immunocompromised, making them highly susceptible to various fungal infections that normal healthy individuals with strong immune systems would not get. So in that case, it would be considered an opportunistic mycosis. Athlete's foot is an infection of the keratinized tissue in the foot, so this would be considered a superficial infection. And eumycetoma is a rare fungal infection that typically only occurs in tropical areas, like South America, Africa, Asia, but it targets the deeper layer of the skin and leads to these infectious masses, predominantly within the foot. It does cause the infection beneath the keratinized tissue, so that would be an example of a subcutaneous fungal infection. So the next type of eukaryotic cell that we're going to talk about are protozoa. So protozoa are eukaryotic microorganisms. They are commonly found in water and soil. They're known for lacking a cell wall, which is different from fungi, because while fungi are eukaryotic, they have a cell wall that is composed of chitin. But protozoan cells, they do not have a cell wall. They are considered other feeders. They live and feed off of other living things in order to obtain carbon and energy. There's relatively few species that cause disease in humans, although we will talk about several species today and elaborate on more species throughout the course of the semester. One thing I definitely want to make sure that you understand is that protozoa can exist in various different stages and go through a cycle. And this is going to sound very familiar to what we talked about with bacteria when I start covering this. But with protozoans, when they're in an active stage, so when they're in a good environment and they have access to moisture and nutrients, they are going to be in their adult feeding stage, which we call a trophozoite. So when they're in a good environment and they're happy and have nutrients, they're going to be in their trophozoite stage. Many protozoa can only exist in this stage, so they can only thrive if they're in a good environment and they're in their trophozoite stage. Some protozoans, though, when they get put into a harsh environment, which for them would be an environment that has no moisture and lacks nutrients, when they get put into a harsh environment, they can go dormant and encapsulate themselves in a cyst. And that cyst will protect them for an extended period of time from nutrient depletion, temperature changes, chemicals, all different types of stuff. Then eventually, once the environment stabilizes, they can go back into their active trophozoite stage where they're continuously feeding and growing and dividing. This should sound familiar because bacteria can do this too. There's different terminology. When bacteria do it and they're active, we say that they're vegetative cells. And when they go dormant in harsh conditions, we say that they're in their endospore form. Same concept here. When protozoans are active, they're in their trophozoite stage. And if they're able to, not all are, but if they're able to, when they get into a harsh environment, they can form a protective cyst to protect themselves for an extended period of time until they get back into a good environment again where they can go back into their trophozoite stage. We can see this happening here with a protozoan known as Giardia. Well, it's Giardia lamblia. It causes the disease that we know as Giardia, which causes vomiting, diarrhea. Humans and animals are prone to getting it. But here this dog is exposed to the soil where Giardia can often be found. So in the soil, the Giardia protozoan are going to be in their cyst dormant form because the soil is dry, so there's not a lot of moisture for them to be in their active stage. So the dog would ingest the cyst, and at that point, the cysts are now in a good environment. So they have moisture, they have nutrients, and they would be able to go back into their active stage, their trophozoite stage, which is what we see here, where they have their flagella out, so they'd be swimming around and growing and dividing and feeding and causing disease, which would ultimately lead to vomiting, diarrhea, cramping. And then when the dog uses the restroom, those Giardia cysts are going to be released in the feces, which would potentially get back in the soil again, and the whole cycle would just repeat itself. But this is just meant to show you when the protozoa are not in a good environment for them, they'll be in the cyst phase where they're protected and dormant. But as soon as they get into a good environment, they will become active where they can continue to grow and divide. So the last few slides is just talking about some different protozoans. So the first is Trichomonas vaginalis, which we see pictured here. Trichomonas vaginalis is a protozoan that can cause a sexually transmitted infection. It represents with urinary tract type signs and symptoms. Typically in men. With females, they would have the UTI type symptoms, but it would also be associated with things like discharge. The primary thing I want you to understand about Trichomonas vaginalis is it's not able to form a cyst. It can only exist in the trophozoite stage. So for Trichomonas vaginalis to be transmitted, it has to go immediately from one good environment into the next good environment, because it's not able to survive in harsh conditions. So Trichomonas vaginalis is typically transmitted through direct sexual contact. If it would get on clothing or sheets, that's a harsh environment because it lacks nutrients or moisture, so it wouldn't be able to survive there. So in order for it to spread, it has to go from one good environment immediately into the next, which would be direct sexual contact in this case. We already kind of looked at Giardia. Giardia lamblia causes the GI infection that we know as Giardia. It is able to exist in a cyst form when it's in a harsh environment, and then activate into the trophozoite form once it gets into a host, which is most commonly animals and humans, where it causes GI distress leading to diarrhea, which will then release more of the Giardia cysts that could potentially make it into another host. So anytime you're working with an animal or a patient that has Giardia, you have to be really careful, because in their fecal matter there will be these Giardia cysts that, if get into your body, could make you quite sick with the infection. One thing that they uniquely have, when you look at them under the microscope, they're very easy to distinguish because they have what we call a smiley face appearance. It looks as though they have two eyes and a little mouth. So if you put these from a stool sample under a microscope, you could very easily tell if someone had Giardia. And the last example is malaria. Malaria is something we'll talk about extensively later on in the semester, but malaria is a protozoan infection. It is caused by a type of protozoan known as Plasmodium. Basically how it works is if a mosquito is infected with the Plasmodium protozoa and it bites you, that Plasmodium protozoa is then injected into your body, where it infects and destroys red blood cells, which can become severe and can lead to severe anemia. So over here we have all these red blood cells, but these red blood cells here in particular that have this dark staining region inside, that is actually the malaria protozoa inside of a red blood cell. And what it's doing is it's feeding off of the hemoglobin in the red blood cell, and then those red blood cells are going to burst and die, and then the patient is going to become severely anemic, which if not treated relatively fast with anti-malarial drugs can definitely be life-threatening.